Good morning, good afternoon, or good evening to you from wherever you're joining us today. My name is Dawn Sievert, and I'm the Senior Science Advisor of the Centers for Disease Control and Prevention's Antimicrobial Resistance Coordination and Strategy Unit. And it is my honor and pleasure to be the moderator for today's webinar. I want to welcome all of you to the second CDC AMR Exchange webinar in our series. Antimicrobial resistance, or AMR, is one of the greatest global public health challenges of our time. It is an ever-changing and increasing threat that has the power to undermine the technological advances and extensive progress made in modern medicine, food production, and life expectancy. The purpose of this series of webinars is to share information on cutting-edge research and impacts in the field of AMR, to drive the field to combat AMR forward by sharing what we're learning and what's working, and to engage partners, practitioners, policymakers, and the public to collaborate across industries, sectors in healthcare, community, and the environment on AMR topics. Our first webinar of the series was held in May and covered AMR as a changed world with discussions around building resilient systems for today and tomorrow. If you missed that one, don't worry. All of our webinars are taped and posted so you can view at your convenience. AMR affects public health on many fronts, and it is increasingly clear that it must be addressed in the environment to control emerging resistance and infections in humans and animals. In today's webinar, the second in our series, we'll be exploring the global threat of antimicrobial resistance in water. We'll cover wastewater and surface water, and we'll discuss the work that's being done to track AMR in various types of water, what important information these activities provide, and how we use that information to better understand the impact on human health and to help identify actions that are needed to decrease the risk of AMR spread. I would like to thank all of you for joining us today, for your interest in this crucial topic, and for taking the time to be a part of these discussions. Now it's my pleasure to introduce you to the four experts on this topic of AMR in water who will be talking with us today. Our first speaker is Amy Kirby, who is an environmental microbiologist and is the program lead for the National Wastewater Surveillance System and the Division of Foodborne, Waterborne, and Environmental Diseases at the CDC. Our second speaker is Rod Donlin, who is a research microbiologist and leads the Biofilm Laboratory in the Clinical and Environmental Microbiology branch in the Division of Healthcare Quality Promotion at the CDC. Our third speaker is Tom Whittam, who is a professor and chair of veterinary preventive medicine at The Ohio State University. And our fourth speaker is Carl Frederick Flack, who is an associate professor at the University of Gothenburg. Welcome to all of you, and thank you for joining us to share your knowledge and thoughts on AMR and water. All right, let's get started. Amy, as I said, I'd like to begin with you, so welcome. First, I'll provide a little more of your background, and for the audience, Amy Kirby is the program lead again for the National Wastewater Surveillance System at CDC. In this role, she is working to understand the infection risk to humans from antibiotic-resistant pathogens and threats such as COVID-19 in wastewater. Amy, as I stated, you lead CDC's National Wastewater Surveillance System, which is helping us understand community transmission of COVID-19. The system builds off of a prior investment to investigate antibiotic-resistant bacteria in wastewater. So can you tell us a little bit about the system and how surveillance of wastewater can help us understand the transmission of resistant pathogens?
Absolutely. Happy to be here. And yeah, so we established the National Wastewater Surveillance System, or NWSS, which we pronounce NEWS. So you'll hear me refer to it that way, in September of 2020. So we're just coming up on our 1-year anniversary of the system being established. And it was established as part of the COVID response to provide community level data on COVID transmission and COVID infections in a community. And the reason we wanted to pursue wastewater surveillance is because it has 4 big advantages. The first is that it captures both symptomatic and asymptomatic infections, because we know that about 50% of people that are infected with SARS-CoV-2, the virus that causes COVID-19, will shed detectable virus in their stool. And this happens whether they have symptoms or not. And it happens in adults and children. So that means what we're detecting in wastewater represents all of the infections or the total types of infections that are happening in a community. Second, wastewater surveillance is independent of healthcare and clinical capacity. So it doesn't matter if people go to their doctor because they're sick, have access to testing, or even if that testing is reported, we can still detect those cases and see the impact of that at the community level through wastewater surveillance. Third, wastewater surveillance is efficient. That one sample coming into a wastewater treatment plant can represent hundreds, thousands, even millions of people in our largest wastewater treatment systems. And finally, it's fast. So wastewater can go from the toilet being flushed to a treatment plant in a matter of hours. And then we collect a sample, ship it to the lab, and we can have that data in hand to use as part of the response within about 5 days of when the toilet is flushed. And that gives us more time to look at that data and use it to prepare responses compared to our clinical data, which we would then see those same trends happening about a week later. We know that our health department partners are using wastewater data to understand community transmission. They're using it to make decisions about things like where to put mobile testing sites, how to allocate resources to different clinical systems in their jurisdictions. And they're using it to forecast cases and hospitalizations, because we know that when we see increasing signals in wastewater, in about a week, we're going to see increasing cases. And about a week after that, we're going to see people reporting to the hospital. And so we're going to see increasing hospitalizations. So we can use that signal to get prepared for those incoming surges. And we've been, in this 1 year, we've been really impressed and surprised with how much news has expanded. We currently have 43 public health jurisdictions that are using CDC funds to support wastewater activities in their jurisdictions. And that's 37 states, 4 cities, and 2 territories that are doing this activity. And we have over 20,000 unique wastewater samples in our data repository as of this week. So it really has expanded very quickly. And we're already working to expand and build upon this investment. And one of our first targets for expansion is antimicrobial resistance. We really think that by looking for genes that confer resistance to especially our last line drugs like carbapenems and colistin, we can use that to identify areas and communities where these resistances may be present, but we haven't detected them in the clinic yet. And once we detect them in wastewater, ideally, then we can go in with increased clinical screening to identify those cases early before they're able to spread and cause larger outbreaks through the community. So we're working on that. We're hoping that we can have this expansion to AR implemented within the next 2 years. So this is going to come very quickly. And as you noted, this really, the foundation of news came from an investment in AR to begin with. So in 2018, we funded a study with the University of South Carolina to look for antibiotic resistance genes and bacteria in wastewater. And that was really foundational to help us understand how you can detect these sorts of disease agents in incoming wastewater. We were also looking at the risk of the workers in those wastewater treatment plants getting exposed to these AR pathogens and looking at their infection risk from those types of exposures. And when in early 2020, when the COVID pandemic started, we recognized that the research platform that had been established by the University of South Carolina to look for AR or AMR in wastewater, we could support them in rapidly changing that to also look for COVID. And so as early as March of last year, they started also looking for SARS-CoV-2 RNA as a marker of COVID in the communities. And that data was really the critical foundation that enabled us to build the system that then became news. So it's really been a nice full circle moment to start with AR, go to COVID, and now we're headed right back to AR.
Excellent, thank you. Thanks for that very thorough overview of news. and for providing us information on the significance that it plays in the AR transmission space, and just the breadth of information that it can provide, and its reach across multiple pathogens. Again, like you said, started on AMR, went to COVID, going back to antibiotic resistance for expansion, and there's so much information that it can provide, useful information for multiple pathogens. So I'd like to inquire a little bit further on this. We know antibiotic resistance has the potential to affect people at any stage of life. And we've already said that it's one of the world's most urgent public health problems. So can you explain how a broader reaching surveillance system like this, like NEWS, aids in our understanding of how AMR and water can have an impact on human health?
Yeah, so I really think about environmental surveillance and water and wastewater-based surveillance as falling into two broad categories. So one is a backward-looking surveillance. So you're using that environmental sample, in this case, wastewater, to understand what disease agents, or in this case, COVID or antibiotic resistance, are circulating in the community that's creating that wastewater, right? So we're looking at it and asking, what do the people that are contributing to this wastewater, what pathogens are they transmitting and circulating around? So it's sort of retrospective surveillance, very akin to clinical surveillance, right? What is already present in that community? But we can also think about environmental surveillance from a forward-looking perspective. So looking at what's in the environment to understand what people may be exposed to. So if we know it's here in the environment, could it cause diseases down the road? So projecting what we think the exposures might be. There is some possibility with that for wastewater, but I think the place where we're really going to see that type of forward-looking surveillance be most useful is in other types of water, like surface water, for example, rivers and lakes, or even drinking water. And we already do some of the regulatory testing for drinking water is really about addressing that forward-looking exposure. For antimicrobial resistance, I think that surface water testing is really going to be key to understand how the environmental compartment helps to disseminate and generate new antibiotic resistance. So when we think about the whole One Health spectrum, right? So we have three compartments: humans, animals, and the environment. In this case, we're talking specifically about water. And it's really important to understand that water is not just a pass-through between animals and humans. Things are happening in that third compartment. We know that antibiotic resistance initially evolved to help bacteria survive in the environment. That's where it came from. And so we need to understand what's going on in that compartment and how it relates to humans and animals to really understand the whole cycle of AR. And so that type of surveillance system can help with that. It can also help us understand what are risky behaviors with respect to water and antibiotic resistance. Is it risky, for instance, to go swimming in your community lake? Is it risky to drink river water when you're going camping? Is it risky to go wading in flood water should you have something like a hurricane in your community? Is it risky to take a shower? What's present in our drinking water? Having that surveillance system in place enables us to understand what those risks are and how they compare to each other. And all of that is necessary to develop better interventions to prevent people from getting sick.
Excellent. Thank you, Amy, for all your insights. We have a clear understanding of the important AMR information that can be obtained through a very broad-reaching wastewater surveillance system like NEWS. But then also, thank you for comparing and contrasting the information that we get from surface water monitoring and surveillance, and knowing the difference between the two, the importance of what's present and what's changing. And again, we know that we always need to know what those sources are and where that antimicrobial resistance and that selective pressure is coming from. So I'd like to thank you right now, and I'd like to focus things down a bit and move the conversation over to Rod. So Rod, welcome. First, I'll provide a little more of your background for the audience. So Rod Donlin leads the Biofilm laboratory at CDC. Through his work, he's investigating the role of biofilms in healthcare-associated infections and is evaluating new methods for the detection and control. So, Rod, we know that biofilms grow on surfaces and that they can contribute to causing infection. Can you explain what biofilms are and how they are connected to the spread of resistant bacteria and fungi in water?
Sure. So firstly, we know that microbial biofilms are complex microbial communities. They're ubiquitous in natural and engineered systems, so we're going to find them pretty much wherever we look. It turns out that these biofilm communities are unique in that they generally describe this process whereby microorganisms associate with and attach irreversibly to a substratum, a surface of some kind. It could be substratum such as a water pipe. It could be tissue, human or animal tissue, and causing infections there. It could be in a wastewater treatment process. Many ways that microorganisms associate with surfaces and ultimately form a biofilm. We know that biofilms also are unique in that the cells produce an extracellular polymer matrix. So that matrix would be generally comprised of extracellular DNA, proteins, and polysaccharides, and it ultimately results in the formation of really what we describe as an architecture, a structure. That structure is important for two reasons. Number one, the cells that grow in biofilms tend to be tolerant towards antimicrobial agents. Interestingly, it's tolerance compared to resistance in that these cells are only phenotypically tolerant. Once we take them out of the biofilm, they tend to be susceptible to these same agents, but they're tolerant toward biocides, disinfectants, and antibiotics. Interestingly also, the slime matrix can protect organisms from, for example, antimicrobial resistant organisms can be protected in the slime matrix. And also it's important because we can see horizontal gene transfer in these biofilm communities. We know also that, interestingly, in the healthcare environment, in acute care and long-term care facilities, for example, the plumbing systems, the hand washing sinks, the sink drains, the associated plumbing piping tend to provide ideal environments for growth of biofilms. It's kind of interesting. It's really the fact that these become like biofilm reactors. They're basically inoculated, if you will, with organisms from the drinking water distribution system and the healthcare environment. And so most of the bacteria that grow in these interesting biofilms in the plumbing systems of healthcare facilities are just aquatic bacteria. They're native aquatic bacteria. Most of them are non-pathogenic, and they're just able to grow under very nutrient-limited conditions of drinking water and form biofilms. And it turns out that the nutrients levels, the shear forces and surfaces are required for biofilm microbial attachment and biofilm formation are met in these systems. So what we have then is a situation where we know these biofilms form in these drains, these P-traps, and in the healthcare facilities. Ultimately, they actually provide conditions or niche conditions for the growth of antimicrobial resistant organisms. That's the cool part. It's pretty interesting. So what you have is a situation where you have these drug resistant organisms that are going to interact with and then colonize the biofilm. And once they do, then they're gonna in some cases, they can go beyond just colonization to persistence and even dispersal from that situation. So we don't really understand for these AR organisms, we don't really understand all the factors involved and the mechanisms involved in their ability to persist. One of the things we have found out that's kind of cool is that we've learned that certain liquid waste that tend to go down the drain in healthcare facilities, for example, Ensure or intravenous fluids, can actually provide nutrients for the growth of biofilms and for the survival of these AR organisms in the biofilm. That's kind of interesting. We know that also we do understand a little bit about dispersal, not much, but a little. And that is that we've done work with colleagues in a university and found that these organisms in the biofilm, for example, the bacterial cells in the biofilm can actually move up the drain. So if you, after the seminar is over, when you go back home, I want you to challenge you to look underneath your kitchen sink, and you'll see what looks like a P-trap. It's a plumbing fixture. And in that fixture, we have it's an ideal, beautiful biofilm reactor. But that's what we see in a healthcare environment as well. These organisms are actually growing in the P-traps. And so what happens is over time, the bacteria tend to move up from the P-trap into the sink basin. And when you run the water, the splashing from the water hitting the drain and the areas that are contaminated with these bacteria tend to disperse organisms into the environment. So we have some data showing that this process actually does occur, and organisms can get dispersed, transmitted into the patient care areas where it could cause problems in terms of colonization of patients.
Wow, so thank you for describing to us the serious role that biofilms play in causing infection and spreading AMR, specifically in healthcare. So I feel like we've learned biofilms are everywhere. We have to be careful what we feed them, and they have incredible strength and structure, and they play a very important role in the transmission of resistant pathogens. So I'd like to then ask a step further. Can you tell us about the innovative solutions that CDC and partners are working on to mitigate these biofilms in healthcare facilities, and how these solutions can reduce the spread of AMR organisms in healthcare and the environment?
Sure, I'd be happy to do that. And well, the first thing we found we really needed to do, there were a lot of case studies out there where, different investigators were in outbreak investigations, for example, finding evidence for these AR organisms colonizing the biofilms in the plumbing systems that we've already just described. So we felt we needed to really start by characterizing the community of these P-traps, right? These plumbing fixtures I've described to you. And so to do that, we collaborated with Dr. Amy Mathers and her colleagues at University of Virginia, and actually collected a couple of P-traps from intensive care unit rooms in a acute care hospital, and actually use culture-dependent and culture-independent methods to characterize the community, to really understand which organisms were growing there and persisting there. It was very interesting, very cool data showing these a very diverse community actually found in these P-traps. So then the next thing we were able to do is build a model. And we do a lot of that at CDC in our lab. It's our lab is sort of a combination of a sort of engineering and microbiology. So we like to build models that simulate the real world. In this case, we wanted to simulate a P-trap, right? So we took this community of organisms that we had isolated from the P-trap and used it to develop a 6 species biofilm community. And then we actually grew the biofilm in a model system we had designed, a model in quotes P-trap. And we're able to actually, these 6 different organisms are all aquatic bacteria, pretty much, mostly from drinking water systems. So we were able to actually allow them to grow in this community, and we were able over a period of time, even over serial transfers, to show that we had established a community that we could work with. So then with that community, what we did is we started looking at, can we add these antimicrobial resistant organisms, these gram-negative organisms that produce carbapinamase genes, can we actually add them to the community of organisms in the biofilm and examine how well they persist, how well they disseminate from that and even amplify in the biofilm. And so we're able to do that with a single organism and show that, yeah, we could actually get it to persist. And actually what I told you earlier about adding these what we call liquid waste seemed to really help to allow them to persist in the biofilm. So then the next thing, of course, was, well, what do we do about this, right? How do we go to the next step? What kind of control can we use? And we knew from the literature that a lot of many different groups had a lot of trouble with these biofilms, especially in outbreak scenarios where they would go in and even go to the point of actually exchanging all the plumbing. and they kept coming back. These organisms kept coming back and biofilms kept forming. So we realized, well, we've got a different sort of a different challenge here. What are we going to do about this problem? And so we had been working for a number of years in our lab on lytic bacteriophage treatment. Bacteriophages are bacterial viruses, and they're very effective. They've been used in the past, maybe up to 100 years ago, after they were discovered, for treating various infectious diseases. They're kind of coming back in level of interest in the U.S., but we had been using them for developing systems where we could coat medical devices with phage and then prevent bacterial attachment and biofilm formation on the device and the laboratory. So we thought, why don't we try that? Why don't we see if we can actually target the pathogens and the AR organisms in the biofilm using bacteriophage? Because that's really what we wanted to do, right? We're not necessarily going to try to blast away and destroy all the biofilms in the system, because they're going to keep coming back anyway. We want to rather target these AR organisms that are the big problem in terms of antimicrobial resistance. And so we actually then went out to our, Amy would appreciate this, we actually went out to our local wastewater treatment plant and collected some untreated wastewater, which is a beautiful source for phage. I mean, you can find whatever phages you want in untreated wastewater. So we got the phages, we worked with them, we purified them. We developed what we call the 4 phage cocktails. We combined 4 different bacteriophages into a single treatment, and using that treatment then and combining it with what's called a non-ionic surfactant. We had some work that I had been done a number of years ago on non-ionic surfactants, something that could help disperse the biofilm to kind of get the phage into the biofilm matrix. And so using that approach, we actually were able to show in our laboratory model that we could in fact significantly reduce this organism, and it happened to be Klebsiella pneumoniae with a KPC gene. Klebsiella pneumoniae, that's carbapenemase, producing carbapennemases. We could actually show in our model the phage would actually work. They would actually reduce significantly this Klebsiella pneumoniae organism while not even doing anything to the other bacteria in the model, because we don't need to. Phages are very, very highly specific for their host organism. So they don't even touch the other bacteria. They just target these pathogens. And interestingly, we were able to show that the phage got into the biofilm. The surfactant helped them to get in there. And the phages, one more thing, the phages happen to produce an enzyme called depolymerase, which is an enzyme which will basically hydrolyze the biofilm matrix. So between the phage's ability to target the matrix and the surfactant, we could actually show that it actually did work and actually significantly reduced the organism. So, well, this is really just, we've published this work, but it's really just preliminary data. We need to scale it up, right? So we have built a laboratory at CDC. We're calling it a sink lab or a sink gallery, where we have these 6 replicate hand washing sinks that are basically the same kind used in healthcare facilities. And what we want to do is scale up our treatment to actually hand washing sinks. And the way that we want to go about this, we suggest going about this treatment is, again, not to kill all the biofilm, but rather just to control these bacteria and keep them out of the sink. Because we know if they get into the sink from the P-trap and the plumbing, that you run the water and they're going to get splashed all over and they're going to get into the patient carrier. So we really want to control that. And so our feeling is we could design the treatment in such a way where we could treat it to keep it down in the P-traps where it should be less harmful. I just want to add a note that this isn't just for Klebsiella pneumoniae. We can get phages for any organism we want pretty much in wastewater. So for example, Pseudomonas aeruginosa, or Acinetobacter baumannii, or any of the gram negatives particularly that carry these genes, we have a way to treat, to develop a phage cocktail for them. And so I think that's probably as much as you want to hear about phage, but I think that there is public health impact of this work. I think it's important because we believe we have some preliminary data that we could build upon to really ultimately lead us down a path of a way to control these organisms in our wastewater system plumbing, which, by the way, it's going to impact what Amy's talking about, because ultimately these organisms are going to flush down the plumbing and they're going to they're going to end up into the wastewater, the community wastewater system. So what we're doing relates to exactly what Amy is talking about. So that's all I have.
Thank you, Rod, and thank you for making that connection. So we understand that biofilms do play a very serious and harmful role in healthcare settings and beyond. You've made that very clear. But it's comforting to know that there's a lot of innovation underway, and do appreciate your explanation of the phage work that is happening to be able to target and specifically mitigate the presence of the resistant pathogens and their risks that they're bringing to the spread of AMR. So appreciate all of that insight. Next, I'd like to slide our discussion from inside healthcare out into the environment of surface waters with a transition over to you, Tom, so welcome. And as with other speakers, I'll first provide a little more of your background for the audience. So Tom Whittam is a professor and chair of veterinary preventive medicine at The Ohio State University. As part of his role, he's studying the emergence and dissemination of AMR across One Health and developing effective antibiotic stewardship programs for veterinarians. So Tom, you've partnered with CDC on numerous projects studying AMR threats across One Health, including investigating the dissemination of carbapenem-resistant Enterobacterales from healthcare facilities into surface waters. Can you review the key takeaways from that work and explain how it can inform future research on the impact of AMR in water on the health of humans, animals, and our environment?
Sure. Thanks, Dawn. Thanks for the chance to be here today and talk about this really important topic. So we did partner on multiple projects. We were really interested in antibiotic-resistant bacteria in wastewater, particularly those that may have originated in large metropolitan hospitals and patient populations. And so that's why we focused on the carbapenem-resistant Enterobacterales, because they're a clinically really important antibiotic-resistant bacteria. And so we had hypothesized that we would find the same carbapenem-resistant organisms in wastewater that were being recovered from patients in their diagnostic submissions. And so once we began surveillance at our local wastewater treatment plant, we did find those, some of those same organisms that were recovered from patients in our hospitals here in Columbus. But in addition, we found that the carbapenem-resistant organisms in wastewater were much more diverse than the ones in the patient populations, more diverse organisms, more diverse genotypes, which told us that these were likely coming from other sources other than just the hospitals. And so that might be other healthcare facilities, maybe nursing facilities, even maybe it's likely to be also healthy colonized individuals out in the community, because we can also find these same resistant organisms in neighborhood wastewater flows, in manhole sewers where there's no healthcare present in the neighborhood. So we believe that they're coming from healthy colonized individuals as well. So that was the first takeaway, is that these clinically important resistant organisms are present in wastewater, but more diverse than the ones in hospital patient populations. So the second was that these resistant bacteria are transported to wastewater treatment plants. And of course, the job of the wastewater treatment plant is to reduce the bacterial loads in the water. But treatment plants, for the most part, are designed to sterilize water that's in wastewater flows, but to reduce the bacterial loads and then discharge the treated water generally into nearby surface waters. And so we found that these important antibiotic-resistant bacteria, the carbapenem-resistant bacteria, they were transported to wastewater treatment plants where the normal treatment did reduce their numbers, but they were not eliminated. Right? And so there were some remained in the fully treated wastewater, and so those resistant bacteria were then discharged into the nearby surface water along with all of the treated effluent. Okay? So we found that was true, that that our ability to recover the resistant organisms was reduced, but we were still able to find them at lower numbers or lower frequency in the treated effluent than in the untreated influent. Also, when we looked at bacterial gene copy numbers or resistance gene copy numbers, we were looking for carbapenem resistant genes, KPC, like Rod was talking about, and also NDM. We found that those were present in the influent and that their numbers were reduced during treatment, but they were not eliminated. So we could still find these resistant copies, these resistance genes in the fully treated wastewater that was being discharged from the plant. Okay, and so finally, as we looked at the surface water around the discharge site for the wastewater treatment plant, we found that these resistant bacteria and the resistant genes were present in the water, and they disseminated around the discharge site, upstream, downstream, and at the site. And as you get further and further away from the discharge site, that the numbers were reduced, our ability to recover the organisms, we recovered them less frequently, and the gene copy numbers were reduced. But we were able to find the same resistant bacteria from the wastewater flow. We can recover them from the surface water itself. We can recover it from the sediment of the river, from the detritus in the river. And I think really importantly, and most surprising to us, we were able to not infrequently recover these from wildlife in the river as well. So we did a lot of sampling of fish in the river around the discharge site for the wastewater treatment plant. And we took just simple vent swabs from live fish prior to releasing them back into the river. And we were able to recover these carbapenem resistant bacteria, the same ones from the wastewater flow, and similar to the ones that are causing health problems in patients in hospitals here in Columbus, present in the vent swabs of those fish near the wastewater treatment plant. And so our ability to find those in the fish was reduced as we got further and further away from the treatment plant. So it appears that these fish are definitely acquiring these antibiotic resistant bacteria in the water and accumulating those, and we were able to find those. So just another opportunity for transmission in the environment and potential human exposure.
Wow, thank you for that in-depth review of your study findings, and for highlighting and tying that entire string together of what we discharge from sources through wastewater treatment to surface waters into the fish and the surroundings and into the environment, just to show how easily these resistant organisms are being spread, and our knowledge of how they get there and why they get there. And this is a very clear line for one of those ways that we're seeing this transmission. And again, so we know that you've presented how this flow moves and with potential sources. And we know that prevention now is going to be the foremost strategy in stopping the emergence and spread of AMR, both organisms and genes. We know clearly that everything is connected, and you drew a perfect picture of how everything is connected. So can you share your thoughts on some scalable solutions to controlling the spread of AMR in water between human animals and within the environment?
Yeah, at this point, that's a real challenge. We don't have really good answers to that question yet, but definitely a lot of really important work going on to try to address that, and such as the surveillance system that's been put in place that Amy's talking about. Some of the research that Rod talked about are really important contributors to that. So there's multiple places where we can potentially implement interventions that could help to address the problem. The first possibility, and certainly that there's some work going on to try to address, is how to prevent the resistant bacteria from leaving healthcare environments in the first place. So are there ways to either contain the resistant bacteria and or treat the resistant bacteria in hospital before it enters the municipal wastewater flow, where it's more manageable, right? And so I think that's and there's various research ongoing to look at different methods for doing that. And there's no one answer to that yet, but that's certainly one place where we could implement effective and potentially effective interventions. So the next possibility would be some additional treatment at the wastewater treatment plants, right? Because that's the job of the plant, is to reduce bacteria. The problem is, as I said before, that most of these facilities, they're not intended to sterilize the water, and really don't have the ability to sterilize it. And at this point, it's not really possible to eliminate the resistant bacteria without eliminating all of the bacteria. And so in wastewater, there's a lot of bacteria, and to sterilize it is not a minor undertaking. And so there's certainly that's a potential site for intervention. And wastewater treatment plants today use multiple methods for reducing bacterial loads. They use ultraviolet light. Right? Sometimes they can use chemicals like chlorination. So there's multiple strategies they use now that do reduce bacterial loads, including the resistant bacteria, but they just aren't set up. They just aren't designed to sterilize water. And so potential to apply interventions there, but at this point we don't have realistic means for implementing those type any interventions at wastewater treatment plants. So that takes us back more to preventing or reducing the selection pressure for resistant bacteria, right? Wherever we use antibiotics. And so one of the ways we do that is through antibiotic stewardship programs. Right? And another thing we need to do is, of course, is infection control practices to prevent transmission. So between infection control and antibiotic stewardship, I think we help to reduce the number of resistant bacteria that the selection pressure that produces those, and reduce their transmission, and so they're less likely to get into wastewater treatment plant flows. So this can and does occur in hospitals all the time. We're starting to see the development and implementation of antibiotic stewardship programs in veterinary medicine, and looking at the ways we can effectively implement those, following models for stewardship programs that are in place today in human healthcare. And so between the stewardship programs and infection control, I think those are really important things that we can do and we are doing now. And then more research on effective interventions and how we can effectively implement those in the One Health cycle.
Thank you so much for that insight, Tom. I appreciate it, and it's comforting to hear that there's some scalable solutions being studied as far as, like you said, the effluent coming out of hospitals before that release and what can be done at wastewater treatment plants. But I also appreciate the fact that you took us back further to the actual source of where this resistance is, where that pressure is, where resistance is coming from, so that we don't look at an end of the line and say, how can we fix it now? But we go all the way back up to that line to the beginning, like you said, with stewardship practices, optimizing antimicrobial use across One Health in humans, animals, and on plants, and then and trying to work towards infection prevention and control, because if we can slow that spread, then there's going to be a lot fewer organisms that ultimately have to be dealt with down that line. So thank you so much for sharing all of that with us. Next, I'd like to take our conversation. We're going to stay in wastewater. We're going to keep talking about antibiotics and even more, and we're going to move to a global perspective with transition over to Carl Frederick. So welcome.
Thank you.
And again, I'll first provide a little more of your background for the audience. So Carl Frederick Flack is an associate professor at the University of Gothenburg, where he studies the role of wastewater on the emergence and transmission of AMR threats in Europe and around the world. So Carl Frederick, from some of the main work that you've conducted globally over the last decade, can you please describe the role of antibiotics in wastewater as a driver for emergence and selection of antibiotic-resistant bacteria, including how risk relates to the levels of antibiotics or selective agents in the wastewater, and how this varies for different types of wastewater and recipients?
Sure. And thank you for inviting me here today. Much appreciated. So as mentioned earlier today, wastewaters and wastewater contaminated surface water may not only be transmission routes for already resistant pathogens, but could also serve as spawning grounds for evolution of resistant bacteria, including the emergence of new resistant pathogens. And why is that? Because many of the resistant pathogens that we encounter in the clinic today, they have become resistant by picking up resistance genes from other bacteria in their surroundings. And these processes, they also include environmental bacteria. And we know that there is a wide diversity of antibiotic resistance genes in environmental bacteria, not yet seen in pathogens. So these could serve as a huge source of new resistance genes for the pathogens. And we also know that transfer of resistance genes between bacteria can be induced by antibiotics. But maybe even more important, antibiotics can be key players when it comes to the maintenance of the bacteria that have picked up new resistance genes by providing a selection pressure that favors the resistant bacteria. And in wastewater and wastewater contaminated environments, we find both pathogens and a wide diversity of environmental bacteria. And in the wastewater, we also, as we have heard, we find various antibiotics, which then could serve as drivers for the evolution of new resistant bacteria. And higher levels of antibiotics provides a higher, stronger selection pressure, which is more favorable for the resistant bacteria. So where do we find the highest concentrations of antibiotics? The highest wastewater concentrations. They have been observed in wastewater from antibiotic manufacturers. In India, for example, Joachim Larsson and co-workers did already in 2007 show concentrations of ciprofloxacin of up to 30 milligram per liter in discharges from antibiotic manufacturers that were released into the neighboring river. And this is an extremely high concentration. It's actually 10 times higher than what you can find in the blood of a patient being treated with ciprofloxacin. So there's no doubt that these levels of antibiotics, they promote and select for antibiotic resistant bacteria. And although large parts actually of our antibiotics that we use, they are produced in India and China, there have been reports of discharges of antibiotics at really high levels from antibiotic manufacturers worldwide. So this is in many ways a real global problem. So even though here then I mentioned that the highest concentrations can be found as a result of emissions from manufacturers, the most widespread presence of antibiotics in wastewater and surface waters is a result of excretions from humans and animals being treated or administered with antibiotics. And when we humans excrete antibiotics, they will end up in wastewater systems, if such are in place. And therefore, you can, in this case, expect the highest wastewater concentrations in countries with the highest antibiotic consumption, although water use and other parameters also play a role here, of course. But even though antibiotics in municipal wastewater, in many cases, have been measured at concentrations that in lab experiments have been shown to select for antibiotic resistance, I would still say that there is a lack of studies providing conclusive evidence that selection actually takes place in the municipal wastewater. I don't say that it doesn't happen, but we are still lacking studies, I would say. But there are other types of wastewater. Compared to municipal wastewater, many antibiotics are generally found at higher concentrations in, for example, hospital wastewater. since consumption of antibiotics per person is higher in a hospitalized population. Again, many studies have concluded that selection is going on in hospital wastewater based on measured antibiotic concentrations, but often without direct evidence. But I would say that we took one step closer in a recently published study by showing that when bacteria were exposed to the complete chemical mixture of Swedish hospital wastewater, including all the antibiotics there, it clearly selected for antibiotic-resistant bacteria, and especially multi-resistant bacteria. So to summarize and generalize, one can say that the risks for promotion of resistance, they are highest with wastewaters from antibiotic manufacturers, and that risks are higher with hospital wastewater than with municipal wastewater.
Great, thank you. So thank you for clearly explaining the significance of antibiotics in wastewater, the detrimental role they clearly play in driving AMR, and then again, explaining how that risk can vary, as you pointed out, with manufacturers and hospitals being lead drivers of the resistance and the antibiotics that we see in wastewater. So I'd like to ask if you could please expand in more detail on the idea that wastewater analyses could serve as a resource-efficient complement to a traditional clinical surveillance globally, and highlight how such a surveillance system would be of particular value in low to middle income countries and why it's so urgently needed.
Absolutely. I should start by saying that clinical surveillance of antibiotic-resistant bacteria is one of the cornerstones in our fight against antibiotic resistance. Such surveillance provides information about resistance prevalence in pathogenic bacteria, which then can serve many purposes. Not least, it can guide prudent and effective empirical antibiotic treatment of infections. And the most important factors for informative surveillance are that the sampling reflects a large number of people and that it's conducted on a regular basis in order to adapt treatment guidelines to an ever-changing resistance landscape. And regular sampling of many individuals is costly, cumbersome, and requires considerable infrastructure. And these are major reasons behind the limited and often complete lack of clinical resistance surveillance data in many low and middle income countries. And here wastewater monitoring could be one part of the solution. As mentioned here earlier by Amy, a single sample of untreated wastewater contains excreted bacteria, including many important pathogens from a large number of individuals. And therefore, analysis of wastewater could provide a resource-efficient surveillance system for antibiotic resistance, which should be of particular value for low-income countries. And we have, in 2 recent studies, isolated E. coli from wastewater and subjected those to susceptibility testing, and showed that resistance rates in wastewater E. coli correlate very nicely with resistance rates in clinical E. coli, isolated from blood and urine samples from the corresponding human populations. And these studies, they were performed in Europe, where we have access to relatively good clinical resistance surveillance data for comparison. But at the same time, these studies favor the idea that it should be possible to predict the clinical resistance situation based on wastewater analysis. And based on this, we are right now in the process of performing similar wastewater monitoring in several sub-Saharan countries in order to get a picture of the resistance situation in areas where clinical surveillance data is very limited, but urgently needed. And I also would like to say that there are surveillance initiatives where genetic analysis without the prior isolation of bacterial isolates have been applied to wastewater samples in order to monitor antibiotic resistance, not least the global sewage surveillance project COMPARE, coordinated by Frank Orestrup at DTU in Denmark, where metagenomic sequencing is applied to wastewater samples. And that type of powerful analysis can, among other things, provide levels of very many antibiotic resistance genes in the wastewater samples. And the metagenomic approach is an attractive option for the monitoring of the overall trends in antibiotic resistance. But there are also some limitations, I would say. Because without isolating the bacterial strains from the samples, there will be uncertainties which types of bacteria, which bacterial species that are carrying the detected resistance genes. And it will also be challenging, or it is challenging, to detect resistance that are due to chromosomal mutations by this approach. And these things, they limit the opportunities to say something about resistance prevalence in specific species of clinical concern. So in relation to wastewater monitoring, analysis of isolates of targeted pathogenic species, I think, is key in order to be able to provide information that in the end could possibly guide empirical treatments.
Thank you, Carl Fredrik, very much. So I really appreciate you sharing the details of your work. And I think we all now better understand the importance and further importance of wastewater surveillance to track the levels of antibiotics and the spread of AMR at the global level. And thank you for pointing out that the difference in that need of what clinical surveillance can provide versus wastewater surveillance, and knowing that where clinical surveillance is lacking, oftentimes in low to middle income countries, the wastewater surveillance can actually provide very helpful and useful information. And with your work showing that the matching resistance of what can be seen in the community through wastewater surveillance and how it matches closely with what you'd be seeing in hospitals. Also appreciate you pointing out differences between looking at overall resistance in genes that are present versus then finding those resistance genes and relating them directly to a specific pathogen and knowing that they are coming from those specific pathogens. So really appreciate all of our speakers. And I'm excited now to move to our next round of questions. So we have time to expand on the information we've heard so far from our experts. And I'd like to pose some additional questions for all of our speakers to consider and weigh in on as they wish and as they have thoughts. So the first question that I have is one that we hear asked often. And so I know our audience is likely also curious. I realize that this is the space which we have the least definitive answers, but I also know a lot of work is underway in this space. So if you wouldn't mind sharing some of your thoughts, my first question is, what is the risk of antibiotic-resistant pathogens, genes, and antibiotics in surface water and wastewater? And Tom, you started talking about this a little bit. So as a follow-up to the information you already shared, if you wouldn't mind starting, if you have some insights, I'll start with you.
Sure, thanks, Dawn. There are a lot of potential risks, and we've already talked about some of those. I think some are obvious to us. That there's additional opportunity for transmission once antibiotic-resistant bacteria disseminate in the environment. We as individuals come into contact with surface waters for a variety of reasons, commonly for recreational activities. But being in veterinary medicine, I'm also aware that it's not just us, but it's also our pets that can be in contact with the surface waters and be exposed to these resistant bacteria. And we can see similar health problems in animals, and that they can be a source of transmission back to humans as well. We have the risk of direct exposure to the surface waters that have these resistant bacteria. But a specific risk that I'm really interested in that hasn't been documented yet, but certainly looking at the possibility, is that generally downstream from wastewater treatment plants, at some point there are farms in animal agriculture. And I think one of the risks that I'm really interested in and concerned about is the possibility that clinically important antibiotic-resistant bacteria that originate in human healthcare, go through the wastewater flow into surface waters, might flow downstream and in some way be introduced into farms, into animal agriculture. And at that point, we have additional potential risks. Number one, the risk to the health of the animals on those farms and to the individuals working on those farms, but also the possibility that those resistant bacteria could experience selection pressure, be amplified on farms based on the antibiotics that are used in agriculture. And if that occurs, then there's the potential risk of foodborne transmission of these bacteria back to people. So we haven't documented that, but that's a potential risk that I'm really interested in and concerned about, and one that we're researching right now.
Thank you. Thanks for pointing out that concern, and again for tying a clear line between that flow and potential transmission. So now I'd like to check with other speakers, starting with you, Amy. Is there anything that you'd like to add?
Yeah, I think one of the things that I always like to really clarify for people when we start talking about risk is that when we think about antibiotic resistance in water specifically, although you could think about this for the environment overall, there's really two different types of risks that we want to be able to measure and estimate. The first is this direct infection risk that Tom clearly outlined. So there is a resistant pathogen in the water and you get exposed to it by swimming in the water, drinking the water, otherwise recreating in that water, and get a resistant infection with that specific pathogen that was present in the water. That is not trivial to estimate that risk. I don't want to say that, but it is something that we have all of the a lot of experience doing. So we understand how to estimate that kind of direct infection risk from all different kinds of exposures, including water. So we're still working to get all the data we need to get those risks estimates really solid, but we know how to approach it because it's very similar to what we do for other kinds of infection risk. The kind that is more unique to antibiotic resistance is this less immediate indirect risk, which is that what Carl Frederick alluded to, that you have potentially a pathogen in the water that is susceptible to the antibiotic, right? So it could be treated, and then you have some other source of that resistance gene, whether it's free DNA in the water or an environmental bacteria that carries that resistance. And they can actually transfer that gene to the pathogen. And so now in that water system, you have created a resistant pathogen. And then you could have that direct exposure risk downstream. And estimating the likelihood of that gene transfer happening in water and then resulting in that direct exposure risk down the stream, we really don't have any other systems to look to base that on. And so estimating that risk is really, really a challenge for us. And what is the time on that? Is this something that happens in an hour? Is it something that happens in a month? Is it something that happens over the scale of years? It's probably a little bit of all of those, but we really don't have good systems to ask that question. And we still need the data, just like the direct risk. We need the data to be able to address that. So whenever I get into these risk questions, I always want to really clarify which one we're talking about because we need to tackle them separately.
Excellent. Thank you, Amy. And thank you for pointing out those differences and for really clearly defining them. Rod, how about you? Is there anything you'd wish to add?
Yeah, there is actually, and just based upon just what Amy has just said about horizontal gene transfer, it's pretty well known that in biofilms, we see elevated rates, enhanced conditions for the horizontal transfer of mobile genetic elements, either through conjugation, transformation, or even transduction from phage. we can see that happening. So essentially, and I can't tell you anything about the risk, but I think there's potential for these transfer these resistance genes to different organisms, including a lot of these aquatic bacteria I mentioned to you earlier that we see from drinking water. You put them all together in this microbial community, it's pretty inevitable you're going to see these genes move around to other organisms. And then, of course, they're going to get into the wastewater system. They're going to be dispersed. And that could be a way, especially in these, what I talked about, the P-trap, these environments where you have high concentrations of organisms and you have physical proximity. So we know, for example, that conjugation occurs at a much higher rate in a biofilm as compared to among planktonic bacteria. So for example, we did a study a number of years ago where we were looking at different gram-negative bacteria, and we could see conjugation occurring within hours. Say we'd start seeing transconjugates within 3 or 4 hours of the time we added one organism to the other in a biofilm. So I think there's a potential there. And then you have this whole range of different aquatic bacteria that could be susceptible to this process.
Thank you very much, Rod. Okay, so next I would go to Carl Frederick, and I'm going to go to him, but I'm going to switch questions. So this one's going to be more directed towards the information that you already shared with us, Carl Frederick, to have you start. So what are innovative and scalable solutions for mitigating water pollution from human-driven causes of AMR in low and middle-income countries?
It's muted.
Are you on mute, Carl Frederick?
Sorry for that. Sorry very much.
Oh, thanks.
Yeah, I don't know if it's innovative, I said, but I would say that the most important thing in order to decrease environmental transmission of pathogens, including the resistant ones, between people is to make everyone has access to clean drinking water and safely managed sanitation services. And that also include basic treatment of the excreta and the generated wastewater streams. And this is absolutely not the case today globally. It's estimated that about half the human population today lack safely managed sanitation services. And in many low and middle income countries, these numbers are significantly higher. So that is definitely, I would say, top priority. When it comes to decreasing the risks for evolution of and development of resistant bacteria and emergence of new resistant bacteria in the environment, it is the antibiotics that and other selective agents that should be targeted. And here one should know that basic treatment is often not enough to remove antibiotics from the wastewater. Many antibiotics just pass through these treatment processes unaffected. So more advanced treatment is often needed, such as ozonation and active carbon, for example. And these are rather expensive interventions. So I think prioritization is probably needed. And based on current evidence base, I would say that one of the first goals would be to regulate discharges from manufacturers, ideally through defined discharge limits and emission control programs supervised by public institutions. Because it's surprising to many, but there are actually no specific discharge limits for antibiotics in place anywhere.
Well, thank you, Carl Frederick. And thank you for, no, we can't talk about AMR and water without saying the main point, and I'm so glad that you said it, the importance of clean water and good sanitation. So thank you for stating that clearly. And then also pointing out, the need for a watch over antibiotic use, wastewater treatment plant improvements, like you said, things, there aren't limits set, and it seems like there's a lot of places then where improvements can be made. Our other speakers also have global experience that they didn't have an opportunity to talk about with their topics today. So I'd like to open it up for others who would like to add to this, as I did focus it more globally. Tom, I'll start with you. Is there anything you'd like to add?
Yeah, just add to what Carl Frederick said. I think that in the low and middle income countries, they can have very different healthcare delivery systems than we have, different access to antibiotics, but they still have the need for some of the basic interventions that we are trying to implement as well. And I think those, such as antibiotic stewardship programs and infection control programs, they need to be able to develop programs that are appropriate for their own healthcare delivery systems and are effective in their environments and their systems, and that can reduce the selection pressure that results in the antibiotic resistant bacteria being disseminated into the environment. So I think for us to look for ways that we can support the development and implementation of antibiotic stewardship programs and infection control programs that are appropriate in those diverse healthcare delivery environments, I think is a really important thing that we can do.
Thank you, Tom. Amy, is there something that you'd like to add?
I would add to the chorus of step one is to make sure that everyone has access to safe drinking water and safely managed sanitation. That's really going to be the single biggest thing that will give us a big improvement in water associated antimicrobial resistance globally. It is a huge problem. The one thing I would add is when we're thinking about particularly instituting sanitation systems and wastewater systems, we really need to think about workforce development as well as the hardware of the system itself. Because we've seen repeatedly when these systems are in place, but they're not operated correctly, or there's no plan in place for sustainable operation for the whole life cycle of that system, they don't work. They don't do any good. They might as well not be there. So we really need to be thinking about that sustainable, robust workforce development and supply chain for parts and equipment as we're developing these systems and starting to build the sanitation capacity in these low and middle income countries.
Thanks, Amy. Thanks for that important additional insight. And Rod, I want to check with you. Is there anything you'd like to add?
Yeah, I'd suggest that we do need to focus on infection control. So in the healthcare facilities, especially in these lower income countries, I think that just even focusing on basic infection control in the healthcare environment. So in other words, in the patient care areas, if we do a better job of avoiding dumping things down the drain that don't need to go there, that could help. Using disinfection of the sinks and the sink areas on a regular basis, I think that could help. That could help to reduce the loading of the wastewater plumbing system with organisms that colonize the patients and therefore keep it out of the wastewater stream. I think that could be helpful.
Great. Thanks so much, Rod. Okay, so I'm doing a time check. We have plenty of time. So I have another question and we don't even have to rush through it. So I'd like to hear from all the panelists on this one. As we've been discussing, antibiotic resistance in water is an issue across One Health, with drivers from and impacts on humans and animals and across countries. So how do we approach this issue holistically so that we're addressing it across all of these areas, knowing that one impacts the other? So I'm going to start with and go in the order of the way we had our speakers talk today. So Amy, I'll start with you.
Sure. So, I'm a scientist at heart. It always comes back to data. So I think what we really need to do is get the data to help us understand how these three compartments, the human, animal, and environment, are interfacing with each other, and how AR pathogens and non-pathogens, those environmental bacteria, are behaving and changing, importantly, as they move through each of those compartments. And the way we do that is with surveillance systems in each of these compartments that provide data that is comparable across the compartments, right? Because what we often end up with in science is data collected one way in this system and data collected, a slightly different way over here. And so then when we try to compare them, we can't quite make the comparisons that we want because we're comparing apples to oranges. So as much as possible, we want to think about these surveillance systems so that we have all three of those compartments as apples to apples as they can be. They might be different types of apples, but at least let's keep them all apples. And we already have systems in place for, at least in many countries, we already have systems in place for surveillance in humans, so clinical surveillance of AR. We have many systems in place for surveillance in animals and retail meat products. But we're looking at building a system for environmental AR. And when we do that, we need to think from step one, how is the data generated in this system going to be able to be linked to those animal and human systems? One of the ones that I think about a lot is the National Antimicrobial Resistance Monitoring System, which we call NARMS. So that's an interagency AR system that looks at antibiotic resistance in foodborne infections. And so we look at that in humans, we look at it in animals, and we look at it in retail meat products. And we want to think about, can we add an environmental component that will enable us to leverage those systems that are already in place for humans and animals to really look at what's going on in that whole cycle. But we need to think again from the beginning, what type of data are we generating? And is it going to be the type of data that is apples to what we're already getting? So really taking that holistic approach from the very beginning, instead of worrying about it at the end when we want to do these holistic analyses.
Excellent. Thank you, Amy. Next, Rod, I'd like to go to you. Are you on mute, Rod?
Oh, no. Can you hear me?
Oh, can hear you. Yes, now I can hear you.
Yeah, right. Okay, yeah. So I just would like to tag on to what Amy has just said about surveillance. I would concur that that's very important. We're actually doing a project right now in our group at CDC on examining wastewater surveillance from nursing home facilities as a way to kind of predict or look at associations between what's in the wastewater and what's in the patient population, the resident or the staff worker populations in terms of their COVID-19 infection rates. But we're also, as part of that study, exploring the use of the same approach, but for collecting DNA that we could use to characterize antimicrobial resistance elements in the wastewater. And ultimately, we hope to really scale that up into a surveillance system for nursing homes and other acute care or long-term care facilities. So I think that's one approach. And Amy's mentioned, I think it's really important that you understand that you have a system for data collection so that you can compare what you're seeing in the wastewater systems to what you're seeing in for example, the influent coming in from drinking water systems into healthcare facilities. So I think that's one I guess, approach that I think is important. The other is, I think that we can also look at inside of the healthcare facilities. And what I mentioned to you earlier about using, collecting samples from P-traps, I think there's ways we can develop that into a surveillance system that we could use to, in fact, look for the presence of these organisms and actually target, so that way it allows us to target patient populations actually in the facility in terms of rooms or levels or areas within a building, and get a good sense of how that contributes to what's coming out of the wastewater system into the larger community wastewater treatment system.
Thank you, Rod. Excellent. And next, Tom, what do you think?
Yeah, so I just want to add about the importance of the One Health component here. We've been talking about water and wastewater and kind of the environmental component, but I think we all recognize that this is a One Health problem. Animals, humans, and the environment are all interconnected, and it's really true with antimicrobial resistance. And I think that one of the places we can do a better job is to help with the communication of healthcare providers across the different components of the One Health spectrum. So what we see is the difficulty of clinicians, prescribers, those who are writing antibiotic prescriptions, the potential impact that has beyond just their patient. Right? And so the difficulty for physicians to know what veterinarians are doing, and veterinarians don't necessarily interact as much as they should with pharmacists, and dentists are maybe often and using antibiotics in their own ways. I think the opportunities for prescribers, the frontline clinicians who are prescribing the antibiotics and dispensing the antibiotics, to interact and understand how antibiotic use in their own area impacts others across the One Health spectrum is a really important thing that we could do a better job of.
Thank you, Tom. I really appreciate your insight. Carl Frederick, what are your thoughts?
Yeah, I very much agree with previous speakers here. And I think one of the most important things is that we need more surveillance data, actually. And very important then is, as pointed out here earlier, is that the same antibiotic resistance markers are studied in the 3 One Health sectors. And I would like to mention here one example of such an initiative, and that is the WHO's Tricycle project, where ESBL producing E. coli are studied in human, animal, and environmental samples in parallel. And although the scope here is relatively narrow, it's definitely a step in the right direction. And I would also like to highlight one thing that is important, but not so trivial to study. And that is actually the direction of transmission between different sectors. Because it's quite important to know in which direction things go in order to design the most efficient interventions. I don't have a good answer on how to do it, but again, frequent surveillance data at least would help.
Thank you. Thank you, Carl Frederick. And thank you all speakers. And I really appreciate your wrap-up comments on this last question. I appreciate that you know, in these last number of questions, you've mentioned the importance of clean water and good sanitation. You've all tied that One Health importance together, that everything is connected, and that, you know, humans, animals, plants, the environment, the water topic, you know, we see how so much of what we're doing, our antibiotic use and our discharges, if it's, you know, if it's from our homes, our building structures, it's from our hospitals, it's from manufacturing, it can be runoff, it can be seepage, but that is all going into our water, and we don't see it. We visually don't always see it. And so it's easy not to realize the huge impact that what we do is impacting AMR in our environment and in our water. I appreciate that the detail that all of you have shared to explain the difference to us about wastewater surveillance and surface water surveillance. We talked domestic, we talked global, we covered those levels. The importance of biofilms and the fact that they are there, and as Rod described, they're always there. We have to deal with the resistance components with those pathogens, but there's always going to be biofilms inside our pipes, inside the structures that we live in and that we work in. So this has been a deeply insightful and informative webinar. Our speakers, again, have reemphasized that the environment, specifically water, can be a major reservoir of AMR pathogens and genes that can impact human health and impacts the health of everything, of the rest of the environment, of humans and animals. Contamination of water with antimicrobial residues, resistant organisms, and resistant genes, again, can occur through multiple pathways. It's been made very clear through all of the discussion we've had today. Water resources can be contaminated through seepage into groundwater and runoff into surface water. and is affected by antimicrobial use in humans, animals, and on plants, and by waste management practices that vary around the globe. We know that we have a growing portfolio of knowledge, and we're making great strides. We've heard from our speakers so that there are solutions. There's a lot of work being done. We know that some of the main things that we can do just overall is antimicrobial stewardship and infection prevention and control, and knowing what we are dumping into our water systems. I think Rod had said it when he said, be careful what you're putting into the systems. We know that we aren't supposed to be flushing drugs and chemicals down drains, because now we know that they're not going away. They're staying in our water and making their way through to wastewater treatment plants, which don't remove everything to go back out into surface waters. So we have this growing portfolio of knowledge. We're making great strides. We're using the information we're gathering to track AMR and to respond with containment and improvements, but we still have many unanswered questions, and that also has been made clear today. So we have a lot of work that we have to do around the globe, and we have a lot of problems that need to be fixed. So I want to express my sincere thank you to all 4 of our speakers. It's been an absolute pleasure to discuss this important topic of AMR and water with you. And we appreciate all of the information and insights that you've taken the time to share with us. I also want to thank all of you who have joined us to be a part of these conversations. Your time and interest highlight just how vital it is for us to understand the risk and impact of AMR in water to human health, the health of our communities, animals, the food we eat, and the environment as a whole. Following today's webinar, please watch for a survey to provide your feedback on the second webinar in our AMR Exchange series, and for announcements on this taped posting, as well as for our next webinar announcement, which will be held, which when we do hold, it will be the 3rd in our series. So I'd like to end by thanking all of you and wishing you a good morning, good afternoon, or good evening. Thank you very much for joining.